Good morning, students. In the chapter Conservation of Plants and Animals, the next topic that we will be studying today is conservation of biodiversity. So, what is conservation? Conservation is the protection, preservation, management or restoration of wildlife and natural resources. Humans have been directly or indirectly being dependent on biodiversity for their sustenance to a considerable extent. However, increasing population pressure and developmental activities have led to large-scale depletion of the natural resources. So, there is an urgent need not only to manage and conserve the biotic wealth, but also to restore the degraded ecosystem. What is a biodiversity? Biodiversity is the variability among living organisms from all sources including terrestrial, marine and other ecosystems of which the organism is a part. It includes diversity within the species and between the species. The objectives of conservation is that it has three specific objectives. What are they? They are to maintain an essential ecological process and life support system. The next is to preserve the biological diversity. And the next is to ensure that any utilization of species and ecosystem is sustainable. Now we shall study the types of conservation. There are two main types of conservation, which is in-situ conservation and ex-situ conservation. In-situ conservation is an on-site conservation or the conservation of genetic resources in natural populations of plants or animal species, such as in the forest. While ex-situ conservation is the preservation of components of the biological diversity outside their natural habitats. This involves conservation of genetic resources as well as wild and cultivated species. So the conservation of biodiversity is of two types, in-situ and ex-situ. Given below are the types of in-situ and ex-situ conservation, which we shall see one by one. Under in-situ conservation, the methods that are followed are together called as protected areas. There are three types of protected areas which are set up. The first is national park, the next is wildlife sanctuaries, and the third is biosphere reserve. First, we shall study about the national parks. A national park is an area which is strictly reserved for the betterment of wildlife, where activities like forestry, grazing and cultivation are not permitted. In these parks, even private ownership rights are not allowed. Their boundaries are well marked and circumscribed. They are usually small reserves spreading an area of 100 to 500 square kilometers. Given in this picture are some of the national parks which are set up in India. The next type of protected area is wildlife sanctuaries. A sanctuary is a protected area which is reserved for the conservation of animals only. And here, human activities like harvesting of timber, collecting minor forest products and private ownership rights are allowed as long as they do not interfere with the well-being of animals. So here, human activities are allowed until they do not interfere with the well-being of animals. The boundaries of sanctuaries are not well defined and biotic interference control is permitted. Example is such as tourist activity. Given in this picture are some of the wildlife sanctuaries that are set up in India. The next type of protected area under in-situ conservation is biosphere reserves. It is a special category of protected area where human population also forms a part of the system. They are large areas of usually 5,000 square kilometer. A biosphere reserve has three parts, which is a core, buffer and transition zone. The core zone is the innermost zone and this is usually undisturbed and legally protected area which is meant only for the protection of wildlife. The next is the buffer zone which lies between the core and the transition zone. Some research and educational activities are permitted in the buffer zone. And the third is transition zone. It is the outermost part of the biosphere reserve. Here, human activities like cropping, forestry, recreation, fishery and other activities are allowed. This picture clearly depicts the zones of the biosphere reserve. There are three main functions of the biosphere reserve, which is conservation, to ensure the conservation of ecosystem, species and the genetic resources. The next function is for the development, which is to promote economic development while maintaining cultural, social and ecological identity. And the third main function is scientific research, wherein to provide support for research related to monitoring and education. The next type of conservation is ex-situ conservation, which includes seed gene bank. These are cold storages where seeds are kept under controlled temperature and humidity for storage. And this is the easiest way to store the germplasm of plants at lower temperature. Here, the seeds of the plants are preserved under controlled condition, which is in minus temperature, so that they remain viable for a long duration of time. And the next method of ex-situ conservation is gene bank, where the genetic variability is preserved by gene bank under normal growing conditions. These are cold storages where germplasms are kept under controlled temperature and humidity for storage. This is an important way of preserving the genetic resources. Given in this picture depicts that the genes are stored under their growing conditions. The next method of ex-situ conservation is cryopreservation, where cryo means cold and preservation is to preserve. So cryopreservation involves cold preservation. It is a process which is used for preservation of the biotic parts. This type of conservation is done at very low temperatures in liquid nitrogen. So the temperature used here is usually minus 196 degrees Celsius, which is very much below the freezing point of the parts. The metabolic activities of the organism are suspended under such low conditions, which can be later used for research purposes. The next method of ex-situ conservation is tissue culture bank. Tissue culture is the growth of tissues or cells on an artificial medium. The tissues or the cells are separated from the parent organism and they are used as the explants. It allows the conservation of germplasm in a protected environment and under aseptic conditions. Given in this picture are the plantlets or the explants which are grown on the artificial medium in the tissue culture lab. The next method of ex-situ conservation is setting up of botanical gardens. A botanical garden is a place where flowers, fruits and vegetables are grown. The botanical gardens provide beauty and a calm environment. Most of them have even started keeping exotic plants for educational and research purposes. It also allows human activities such as tourism. Given in this picture is the Botanical Garden which is set up in Ooty. So here we come to the end of conservation of biodiversity by ex-situ and in-situ methods. Thank you, students.